Okay. Good morning, everyone. Today we are happy to have Carles giving a talk. When we have people from the EBD, I mean, we always see them around and we know who they are, but sometimes we don't know some important things about their lives. So I'll talk a little about Carles. So Carles is from Catalonia, and he defended his thesis in the University of Barcelona there in '93, but he actually developed his work of his thesis here at the EBD. So after that, he spent some 3 years at the US in the UCLA, and then almost 10 years in Sweden, so almost Swedish, in the University of Uppsala. And that's when he came back to the EBD. He's been working here for 12 years now, and that's where he's been developing the research that he will present to us today. So thank you very much, Carles.
Thank you very much, Vilà. And well, it feels kind of strange to be talking in front of friendly audiences. I mean, the people and the colleagues. Okay, what I'm going to be talking about is mainly based on a study that was published a few months ago, and I know that some of you already know quite well. This study is the result of a very large collaboration between many research groups, and you will see that during the presentation, sometimes you will see some of the symbols and some of the names appearing of the collaborators. And yeah, although this is the starting point of this project, what I'm going to be focusing mainly, I'm going to be talking as well about the current aspects where we are working on right now, that is continuation of this project. So, how did all this start? Okay, this project is about common quails. The quail, you know well, is a small bird, it's migratory, and we have populations across Europe and Asia that migrate south, go to Africa, India to migrate, and there are some resident populations as well, here marked in green in the map. And we are here. We're working in south of the Iberian Peninsula, and here we have migratory quails. Supposedly we have resident quails that spend here the entire year, and we have flyways, so we have a complex situation with all kinds of quails. And the interest in this project started many years ago. While I was still working in Sweden, more than 15 years ago, a collaborator, Domingo Rodriguez Teijeiro, came to us asking to see if we could try to answer a question. Why are there so many dark quails south of the Iberian Peninsula? He has been monitoring quail populations for over 30 years with a large team of collaborators. They have been sampling quails, thousands of quails, and they noticed that here in the south of the Iberian Peninsula, southern Portugal, they had a larger proportion of these dark quails that seem to have also different morphology. He was even speculating that perhaps they could even have a different migratory behavior because the wings seem different, no? You see, this is a picture that we have taken a few months ago here in Doñana, and well, on the same net, the same day, two very different males, a very light one and a very dark one. This is the more common type across Europe. But here, there are lots of the dark type. So trying to answer this question, when we were in Sweden, we started to genotype some microsatellites. We had a small panel of microsatellites, and well, we ran a structure, we did some analysis, and we saw nothing. Everything was all mixed up, so okay, just natural polymorphism within the population, nothing very relevant. However, Domingo was looking at the data saying, well, maybe, you see, there is a little bit of this type that maybe it's a little bit more frequent. Maybe there are two sympatric populations. And from the population genetics point of view, we're going to say, okay, this makes no sense. How are you going to talk about two sympatric populations that are genetically differentiated? Those would be two different species. This makes no sense. But so we had discussion on what was going on. However, we very recently came to realize that Domingo had not been the first person to suggest that there was something going on with the quails. And in fact, some time ago, we found this. This from an article published in 1963. And look at what it says. It is worth remarking that in his accounts of the quail in his Ornithology of the Straits of Gibraltar, Colonel Irby states that the Andalusian cazadore professed to recognize two kinds of quails, those which are migratory and called criollas, and those which are resident and so named castellanas. Irby remarked that there was certainly much difference in the color of the plumage and on the legs, the criollas being lighter. So 150 years ago, some ornithologists were already commenting that looking at the animals that had been killed by hunters, well, it seems that there are two kinds of quails around here. So maybe what Domingo was seeing, and was a big surprise for us, was something that, well, had been known in some circles for a long time. And in this article, in 63, they were asking Dr. Valverde and Professor Bernis to try to find out what was going on here. So this article, this book, Ornithology of the Straits of Gibraltar, was published 150 years ago. So, and we have been knowing nothing about this since then. Well, because of the insistence of Domingo, we started to work on this topic, trying to see if we could find what was going on. And this started with a small project. In fact, one of these Severo Ochoa micro projects, a €3,000 project that was initiated by Ines, trying to, well, use genomic markers. For us, was a learning experience to use genomic markers, and we would see if that could tell us something. We selected individuals, 8 individuals from these 9 populations, including some of these in south of the Iberian Peninsula, that could tell something about these differences in color in the quails. So we used a genotyping by sequencing approach. So instead of 12 microsatellites, we have 50,000 markers. And then what Ines was doing was trying to see what was the differentiation along the genome. Let's put all the markers one behind the other along the genome and try to see if there is any marker, any area where there are markers that show some difference between populations. Maybe that could help us identify genes associated with pigmentation or something like that. This is what we saw. There was not one peak, but there was this gigantic region in chromosome 1 that had huge differentiation. Suddenly there was a very large block in chromosome 1 that was very strongly associated with the difference between the populations. We had no idea what this was, and because of our experience, our first interpretation was that we made a mistake and there was some sort of contamination, but we could not figure out what kind of problem that was. So, well, we kept searching, and let's try to find out what is going on in this area. So we selected the 8,000 genetic markers that were found in this region of chromosome 1, 8,000 SNPs. And with that, we could try to characterize all the quails. And this is what we saw. This is equivalent to a structure analysis that many of you have used. And we can see that, well, each individual here appears as a vertical bar. And we can see that we have black bars, we have red bars, and some bars that are half and half. This is very weird, very uncommon. We told the computer, okay, make 2 groups, and made 2 groups that were completely black, some other ones that were completely red, and some individuals were half and half. This seemed to suggest that there were 2 different chromosomes, 2 completely different blocks of markers that were inherited as a unit. Some were red and some were black. The red ones, you can see we are representing them with this red dot, could represent one chromosomal type, And you can see some individuals in southern Portugal, in the Canary Islands, that are completely red. And we have some other individuals that appear as black with the other kind of chromosome, and some other individuals that are perfectly 50-50. It seems as if we have, well, two types of chromosomes that are moving around the population. And that was not all. These two chromosomes are not distributed at random. If we look at the frequency of the homozygotes for one kind of chromosome, the black dots, the homozygotes for the other kind, the red ones, and the heterozygotes, the blue ones, we see that it's only south of the Iberian Peninsula, Macaronesian Islands, Morocco, where we have these blue and red types. So they seem to suggest that there was something preventing the mixture of these two chromosomes. What was going on? Our first interpretation said, well, maybe there is an inversion. Maybe there is something that prevents these two chromosomes to get all mixed up. And we have the red chromosome and the white chromosome, let's say, or the inverted and the non-inverted. So this is kind of what we were thinking, that in chromosome 1, we have some individuals that have this block of the chromosome that is inverted. And when that happens, we could expect something like this. You see here we have a blue chromosome and an orange chromosome. The two have the same genes, but in the orange chromosome, the genes B, C, and D are inverted, are in a different order. These two chromosomes, if they are found in the same individual, they can meet during the meiosis, they can recombine, but only if they do some very strange structure. They could pair with each other during meiosis, and when the individual carrying those two chromosomes produces sperm or eggs, would end up with some individuals that are recombinant that would miss many of the genes. So we'll have chromosomes that would not be viable, or the recombining ones. And the ones that have the complete sets of genes, sorry, these two, are the ones that are as they were at the beginning. So at the end, this implies that if there is recombination inside the inversion, the chromosomes would not be viable. So that implies that basically we don't have recombination in the living population. Okay? So we have all this group of genes that are inherited as a single unit. So this was our hypothesis of what could be happening. How could we test if this was the case? We know that chromosomal inversions are very important in evolution. Now that we're starting to have extensive genome data, we start to find more and more studies that show differences like this. We can see that in some species, we can see differences in behavior associated with inversions, like trouts that would be anadromous or resident, living together, but basically not mixing. We can have differences in sexual behavior, in mating systems. We can have differences in pigmentation. All these differences can lead to speciation. Okay? So in recent years, we see that these inversions could be associated to some of these phenotypic changes that we see in populations. Okay. So let's see if we have an inversion. For this, we collaborated with people at the Autonomous University of Barcelona, and basically what they did is to develop two probes, one that would be inside the inversion and one that would be outside the inversion with different colors. And the idea would be to make immunofluorescence, make cytological preparations, and try to see if we have results like this. If we have in some individuals, that ones that appear not to have the inversion, to have the green and red dots very far apart. If they have the inversion, they would be very close together. If they're heterozygotes, the two red dots would be separated from each other. And this is exactly what we see. We could confirm with immunofluorescence that the individuals that do not have the inversion have the two markers very far apart. the individuals that do have the inversion, they have the two markers are very close together, and in those potential heterozygotes or heterokaryotypes, we could see that there are two red dots because they are not together. Okay? So we could confirm that there is an inversion. Okay? So once we know that there is an inversion, let's try to understand what is going on within this inversion. Here comes the work of another PhD student, within the group, Sara Rabagni. What we did was to select 16 individuals and we sequenced the complete genome. We have 6 individuals without the inversion, the ones that we represent with the black area or the dark gray area, and 10 with the inversion. We try to get them from very different populations, from the Canary Islands to southern Portugal, Morocco, Madeira. We have to try to have some diversity, and the same for the ones without the inversion, from Italy to Canary Islands, trying to have as much diversity as possible. And then we did the same. Let's look at the differentiation along the genome between these quails with the inversion and without the inversion. You say, remember the figure that Ines did? So this is what Sarah found. Inside chromosome 1, there is this gigantic area that is very different between the two groups, the ones with inversion, without the inversion. Now that we have the genome sequence, we can measure how big that is. 115 million base pairs. And this is about 12% of the genome, and we know now that it includes more than 1,200 genes. So we have 1,200 genes that are inside a block that does not recombine with other. So we have this gigantic supergene that is evolving independently in one group and the other. 1,200 genes that can be accumulating divergence independently in the two groups of quails. Okay? So, if we look a little bit more carefully, we can look at the genetic variability within the inversion for the two groups of quails. We see that if we ignore the inversion, for the rest of the genome, basically the two types of quails are the same. They show the same variability. But within the inverted region, within this 150 million base pair, we see that these quails that are in south of the Iberian Peninsula and Canary Islands, they have very much lower genetic diversity. Our first interpretation is that perhaps this is telling us that this is the inverted version, that this is the original and this is the inverted, and has had a shorter time to accumulate divergence. Although, at the same time, this is distributed over a very much smaller area, has a smaller effective population size, and is slower at accumulating genetic diversity. But all this led us to think that this, the B version, is the inverted part, no? The one that is restricted in the south of the Iberian Peninsula and in northern Africa and Macaronesian. That is the inversion. And, well, we could think, where is this coming from? Do we have any idea what could be the origin of this? When did this inversion originated? Okay. Well, we could just compare the sequence in the inversion in the two types of quails, and looking at the sequence divergence, we could estimate the time of divergence. And we estimated that the two chromosomal types originated at least 1 million years ago, and could be very much more than that. But the two types, the two chromosomal types originated at least 1 million years ago. It could be even older than the divergence between the common quail and the Japanese quail, the sister taxa that is distributed in Asia, in Japan, China, Mongolia. The divergence could be older than the origin of these two species. Okay, so something happened more than 1 million years ago. Well, what could have happened? We have been discussing two possibilities. The first one is incomplete lineage sorting. More than 1 million years ago, one individual had this inversion, and that remained within the population, within the species, has been evolving within common quails. And just because by accident has been remaining only in this part of the distribution. Against this argument is the fact, well, we see it on the common quails, but not in the Japanese quails. We see it only in a very small area. If it's so old, supposedly we should be able to find this inversion in many other areas, but we do not really know. However, there is an alternative explanation that is very, very appealing. Maybe this inversion, in fact, derives from another species. Another species that has been evolving independently, they interbred, and because the other species has different chromosomal arrangements, well, this inversion has remained within the population, but in fact derives from another species. This has been confirmed in some other cases, in some other inversions, and that are very well studied. None has been as big as ours. This is the largest inversion that has been studied so far. So, well, this could be a possibility, and in fact, well, looking at the distribution, we could think about what other quail species are around here. Well, there are only 3 that could overlap with the distribution in Europe and Africa. The blue quail, sometimes called Coturnix adansoni, the harlequin quail, and the rain quail. Okay, maybe this inversion derives from an interbreeding with one of these species. However, we don't have this data yet. It's something to work on in the future. But it seems that the evidence available so far suggests that this species separated from the lineage leading to the common quail and the Japanese quail very long ago, much more than 1 million years ago. So it seems that maybe it's not very realistic. It seems that probably none of these species could be in the origin of the inversion. However, there is another possibility. Maybe this inversion represents a ghost chromosome. Maybe it comes from an extinct species. And this was a study that was published in 2020 by some paleontologists that were studying quail remains in the Macaronesian islands. And they could see that there were multiple species that inhabited the Canary Islands, Cape Verde, Madeira, Azores. They were morphologically differentiated. They were considered different species. And all of them went extinct with the arrival of humans to these islands a few hundred years ago. There was one species in Cape Verde. There was one in Canary Islands, 2 in Madeira, and in Azores they are still describing a couple more species. So maybe what we see is a fragment of the genome of one of these extinct species. So this is one of the lines that we're working on. In the ancient DNA laboratory, we have bone remains of these species, of some of them, and we're trying to see if we could find any relationship between the genome of these birds and the inverted sequences. But this is ongoing work. Maybe in a year, maybe we'll be able to say something or two. Okay, but let's look a little bit more in detail in this inversion. Okay, I told you that we had the complete genome of 16 birds. Okay, 10 with the inversion, red lines, 6 without the inversion. If we ignore the inversion and we look at the rest of the genome and we try to make a phylogenetic tree, this is what we get: nothing. Okay? We do not see any clusters. Shows that looking at the overall genome differentiation, excluding this fragment, there is no structure at all. We see here close to each other individuals from Italy, from the Canary Islands, from Morocco, from Portugal, all mixed up without any kind of a structure. This is basically what we can expect. Common quails migrate, and they are known, well, they are supposed to do something that is very uncommon for migratory birds. It has been claimed that females, when they migrate and reach southern Spain, for example, they can reproduce here, have their eggs, they have their chicks, and when the chicks start moving around, they keep migrating and move and have another clutch, maybe in Denmark. So we could have, on the same year, siblings being born in Seville and in Copenhagen. So with this big mobility, we cannot expect any population structure. Everything should be all mixed up. And in fact, our collaborators from Barcelona have been monitoring quails for many years. And the idea is that, well, both males and females are in continuous movement. It's very difficult to recapture in the same locality the same bird during the same season. They lay eggs, they have their chicks and move out. And males, not even that, they don't wait that long. 1 week and they are out, okay? Is what the Domingo and company called the movimientos don Juanescos, trying to find partners around the distribution range. So this is then what we could expect. But what happens if we look at the inversion? Okay, we look at the inversion, the tree is slightly different. There is something big going on. This is the ancestral sequence, the non-inverted, and that shows no pattern at all. Those individuals are migrating, are moving, but the other ones, the ones that have the inversion, show a strong structure. Remember, these are the same individuals as before, but there is a different region of the genome. And we can see here that there is geographic structure. Here we have the cluster of the individuals from the Canary Islands. These are the individuals from Madeira. These are the individuals from the continent. There is geographic structure. So we think that this suggests that individuals that inherit this inversion change the migratory behavior. And the individuals that are in the Canary Islands and inherit that fragment with the inversion, maybe they stop migrating and they stay there. And the fragment with the inversion, these 150 megabases, do not move from the archipelago. But individuals that do not inherit that keep moving. This could be just a speculation, and well, but we can try to see if the ecological and morphological data supports this. So we have been compiling morphological data from all these animals, and look at what we have. This is the weight. We have individuals from those 80 that Ines analyzed, that we have the genotype, and they have homozygotes without the inversion, homozygotes with the inversion, or heterozygotes. We see that there are significant differences in weight. That individuals with the inversion, these red ones, here, are heavier. And in the same population, we can have individuals of this type and in this type, here in Sanlúcar, mating, having litters together, but there is a significant difference in the weight of the two types. And the heterozygotes, heterokaryotypes to be correct, are somewhere in between. We could also make an analysis of the pigmentation. We could label, just to simplify, make 6 categories according to the degree of pigmentation on the neck, on the throat, and just count how many individuals we have from each category. And we see that while common quails on most of the distribution are relatively pale, here, the ones with the inversion tend to be very much darker, and the heterozygotes are somewhere in between. And we can even apply this index, the Holinsky index. This is an index that tries to reconstruct the wing shape. No? Low values suggest rounded wings, high values, pointed wings. This is a cartoon, this is not real. This is not how the wings look like. But this is much more efficient for sustained flight, for long migration. And it seems that all these ones with the inversion, these ones that seem that do not move that much, have rounded wings that are very inefficient. So it seems that, yes, there is something in the morphology that makes that individuals that inherit this inversion start to have wing shapes that are not very efficient for dispersal or for sustained flight. Well, this is just morphological analysis. But with each individual that we captured, we could take them feathers. And my quails molt, interrupt the molt, the change in feathers during the year. There are some feathers that are grown in wintering grounds and some feathers that are grown on the summer grounds, on the breeding grounds. So we could look at the isotopic composition and try to see if the habitat seems the same. And this is what we see. This is just for two of the isotopes. We see here the deuterium and carbon. And let's look at the black box. That is the quails without the inversion. There is a big difference between the feathers from the breeding grounds and the wintering grounds, and even larger for carbon. But the other ones do not show any difference. You see? It seems that the individuals that do not have the inversion are spending the summer and the winter in very different areas. Individuals that have one or two copies of the inversion, they do not migrate, or they migrate very little. Okay? So it seems that this inversion affects the morphology, but also the behavior. So, we have then this block of genes that is not recombining, that is affecting lots of many aspects of the life history and the behavior of the quails. Every time I've been making this presentation, the same questions come up. Okay, what is inside this big genome fragment? Well, it's 1,200 genes. There are lots of things. And we have not done yet a very detailed analysis of all the 1,200 genes because we expect that we can find anything there. But from the original publication already, Sarah observed that there are two genes associated with pigmentation that show very clear differences between quails with inversion and without the inversion. So that could be associated with the differences in color. Okay? But things that we are now working on, we are trying to look with more detail into this, and we see that there are other things. If we look at the entire fragment, we could look at SNPs at variable positions within the inversion only, and look which ones are within genes and which ones are responsible for changes in the protein and which ones are synonymous. Okay? So this is a ratio of the non-synonymous to synonymous changes, the NDS. So, and we see that is very much larger in the quails with inversion. What is this? This is telling us that in the quails with the inversion, there is a faster accumulation of mutations that lead to morphological change. It's not that the mutations are accumulating faster, but that they are removed less efficiently by selection. Selection here, in the ones without the inversion, is more efficient because there are many more quails. So selection is removing lots of these deleterious variants. But in these quails in south of the Iberian Peninsula, Canary Islands, we have lots of mutations that are accumulating, that are producing phenotypic effects. And the selection is, well, the population is smaller. It's not as efficient in removing it. Okay? Because the population size is smaller. And also the selective forces may be different. These individuals do not need to migrate. Maybe they have fewer constraints on the body weight, on the wing morphology, on the fat deposits, on the physiology, that for migratory quails, well, they have very much higher limitations. In fact, if we look at the genes that have a very large number of SNPs, and we look at the genes that have larger differences between the non-synonymous and the synonymous mutations, we see a concentration of genes that affect anatomical structure development, more genes affecting morphology. It seems that because of the smaller population size, because of the lack of migration, there is a relaxation of selective forces in the quails that live around here, in south of the Iberian Peninsula, Morocco, Canary Islands, that could lead to faster morphological change, faster divergence. No? And, well, we're still digging into this data, trying to understand what's going on. This is far from done. Well, looking at the genome and having this complete genome data reveals other kind of things. After excluding all this region in chromosome 1, we have found that there are other inversions. There are other portions of the genome that may be affecting the biology of quails. This is one that we have found. We do not have very much data. This is in chromosome 2. This is 50 million base pairs, 50 megabases. It's not associated with the first one. It's not associated to geography. And we do not know what could be the effect. It's something we are looking on. But there are more inversions that could be affecting the diversity of behaviors and population structure of common quails. And in fact, our preliminary analysis suggests that this inversion could have a divergence time very similar to the other one. This may be supporting the idea that both of them originated from the introgression with a different species, from an interbreeding with a different species, not just something that happens within the species. So this is justifying even more the need to analyze these ancient specimens from Canary Islands. Okay, Anna? Thank you. Okay, so this is still work ongoing, lots of things to be done. Just to wrap up a little bit what this is taking us. So we know that there is a big genomic inversion in chromosome 1 that affects a large proportion of the common quail genome, limits the recombination of 1,200 genes, so they are inherited as a single unit, and is restricted in geography. And it seems that this inversion is associated with pigmentation, body morphology, flight capacity, migratory behavior, and is associated also to faster evolution. Okay? And we have seen that there are other inversions in the genome. And, well, this could be a good point to stop, but I'm going to trap you a little bit longer. Because, well, I guess that many of you are thinking, well, you are talking about Macaronesian, you are talking about Canary Islands, about Madeira, you even mentioned Cape Verde. But there are more islands. Sorry, before that. Sorry. If we look at these aspects, before moving to other islands, there is something very important here. We see that there is big differences in the two lineages in quails, and they live together. But does that mean that they are completely fertile? Does that mean that they can interbreed? Does that mean that all individuals have the same fitness? Some collaborators were surprised with that this gigantic inversion could be even viable. Okay? So there are some doubts if there is some sort of isolation. Maybe we're at the beginning of a speciation process. Okay? And this is part of the thesis of Celia, looking at what is going on with these populations. Celia and, of course, Inés have been looking at the quail populations, focusing on Doñana, trying to see all year round We have been capturing lots of quails during winter. Who is there in winter? Do we have only these quails with inversion? Or those are gone maybe just to Morocco, and what we have are quails that are migrating from Northern Europe? Is that really a resident population, what we have in winter in Doñana? And well, if we have the two types of quails, with inversion, without inversion at the same time, do they breed at the same time? Maybe the ones that are resident, they start to breed earlier, and there is some phenological segregation, that some of them start breeding earlier, and well, they keep separated. And well, to have a better understanding of what is going on, we have been monitoring populations in Doñana all year round, from doing field surveys when there was a few degrees below zero, when they were reaching the thermal tolerance for biologists and for quails, and over 40, 45 degrees, and there were quails all the time, no? And that offered also excellent opportunities for the training of people in the animal experimentation. Okay? And yes, we're using masks when we're mandated. I'm saying that because of Eloy. Okay, but this is, we have been talking about Canary Islands and Madeira and Cape Verde. Oops, but what's going on with Azores? We had this data when we prepared the article. We just said, okay, that's too much. That's too complex. There is something else going on. And those are the same 80 quails that we started the study with, that Ines analyzed. We had at the same time some quails from Azores. There already now people are saying that quails are different, have a different morphology, and they call it as a different subspecies. But if we look at that data, we can see that, well, you see, we do something equivalent to a principal component analysis, and we can see here those 80 quails that have, and that we have been discussing so far, the ones without the inversion, the ones with the inversion, the heterozygotes, and the Azores quails. They are completely different. There is something going on there, and also very spread. And we're talking about an island population of resident quails, small, isolated. So what is going on there? So we excluded them from the first analysis because we had no idea how to put them together. Now we have solved what is going on down here. We're trying to understand what is going on up there, and it's something that we hope that Sarah will soon finish. But the first thing is, well, these quails, do they have the inversion that seems associated with the lack of migratory behavior? So we did the comparison between the quails from Azores with quails without the inversions and quails from the Azores with quails with the inversion. And we see that here the big difference still appears and not there. So this seems to suggest that the quails from the Azores do not have the inversion. No? Have a completely different origin. And in fact, if we just look, hopefully, there you go. Okay, if we look at the SNPs distributed across the entire genome, we could do a figure like that. In the top one, you can see, we do an structure analysis, admixture analysis with quails. Here you can see with the inversion, without the inversion, here are the heterozygotes, and we would tell the computer, okay, make two groups of samples, the Azores quails cluster with the ones without the inversion. When we tell the computer, make two groups, puts them as a different group. But the first step is to put them with the quails without the inversion. But if we ignore this part of the genome, that we know it's weird and affects a lot of what we are doing, if we exclude the inversion, this is what happens. With two groups, we can see that the quails from the Azores are completely different from all other quails. Okay? After excluding the area of the inversion, we can see that the Azores quails are something else and do not have the inversion, or at least not all of them have the inversion. So it seems that there has been a loss of the migratory behavior that is completely independent to the loss of the migratory behavior that has happened on the other islands. It's not related to chromosome 1, to the inversion in chromosome 1. So, yes, they are different, but I told you that they were also suggested to be even a different subspecies. Well, we did a clustering analysis, just looking at morphological data, and we saw that when the computer was suggesting to make 3 groups of quails, when putting together continental quails and Azores quails, and one of the groups, number 2, was only Azores quails. And the 3 groups that the computer tend to form, one was very similar to the quails with the inversion, another one with the quails without the inversion, another one only referring to Azores quails. So we can see that Azores quails have shorter wings, are very dark in color, they are very small. In fact, they can fly through the net that we normally use to capture the other ones, and they are also small tarsuses. So we have a completely different lineage that is living these islands and, well, an independent lifestyle. So now we'll stop here just for a moment. What is going on? The project has been involving lots of people, the number of participants is growing, and there are lots of questions that need to be addressed. Does the inversion affect the fitness? Are the 2 types living together and mating together without any restriction? Are in Doñana, can we find the 2 types interbreeding, or is it just an accident? Is there any assortative mating? Are there some individuals that start reproducing before the other ones? Still, what is the origin of this inversion? Can we confirm that it's an interbreeding with an extinct species? That needs a lot of work. Can we be sure that the distribution is restricted to this area? Maybe it's the only area we have looked in detail, and we need to expand the surveys. If these quails do not migrate or migrate so poorly, how have been the different islands colonized? Okay? So there are multiple lines that we are still working. There is a lot of work to be done. There is a lot of people involved in this project, different aspects, and I would like to thank all of them. You have seen the names popping out, and with that, I just would like to thank you, and I'm available for any questions.
Okay, any questions for Carlos? That's a quick one.
Hi, Carlos. Great talk, thank you. I was wondering if when you calculated the divergence time between the inversion and the non-inversion, seeing how there is like signals of accelerated evolution in that area for different reasons, do you think that that inference of that divergence time might be affected by that? And maybe if you look at it like, I don't know, if you're using the genome-wide mutation rate, then maybe there it should be adjusted for a specific mutation rate for the area, and that might affect how long you're going into the past looking for possible hybridization events. I don't know.
Yeah, that is a very good question. And in fact, what I've done here is to simplify a lot. If we go back to this slide, sorry, up there, here, okay? Basically here we compare the genome data for the inverted quails and the non-inverted quails. There is an orange line here. The orange line is an estimate of FST, but over long windows. Each one of those dots represents 10,000 SNPs. The line is an average over 1,000,000 SNPs, no? So it's a smoother line. And you can see that there is like a U shape. That is what you would expect if occasionally there is recombination. What I said is that the recombinants are not viable, but that's an oversimplification, because if there are 2 recombination events inside this region, that could be viable if there are 2 recombination events. And if that happens, that would imply that there is a little bit less diversion towards the center of the inversion. And this could be what this is saying. With this, where I'm going is that we can see that the divergence is higher at the beginning and at the end. So for those analyses, what we did is first to select only in the most extreme areas of the inversion, not the entire inversion, but just the areas to make sure that we're avoiding any recombination. And that would be the first step. The second thing is that do not believe those numbers that I said at all, because we do not have a good estimate of the molecular clock. There have been different molecular clocks, different for chicken, for zebrafinch, for birds in general, and all of them are quite different. What we said is that the divergence is at least 1 million years, could be longer. So this is the lowest estimate that we obtained. And the other aspect that you were saying is also the changes in the rate because of the acceleration of the mutation rate. That is true, but inside these 120 million base pair, or 150 million base pair, there is lots of non-coding sequences. So we expect that these selective forces that can reduce part of the variation would be for non-silent changes and non-synonymous changes. So most of the variation we have taken into account is silent, at least as far as we know. So we don't think that these changes in the substitution rate, or sorry, in the selective forces would translate into big differences in the mutation rate. So we are not worried in that direction. I think it's much of a problem that we don't have any idea about what would be the best mutation rate.
Okay.
So that, and also that we have to restrict the analysis to some areas where we expect that the probability of inversion is even lower. So,
Thank you.
Thank you.
I'm just curious, you mentioned that you were addressing these next studies, so the assortative possibility of assortative mating, but with the data you already have, you probably can do some kind of Hardy-Weinberg test.
Exactly, that is our initial idea. For those that do not understand the reasoning, basically, where we can look at the breeding season to see, okay, who is here? And what is the genetic composition? How many have the inversion? How many do not have the inversion? And we can make a probabilistic analysis and try to say, okay, if they were mating at random, we would expect that many individuals carrying the inversion. This is the reason why we are doing the surveys. And in fact, some reviewers asked us to do this analysis in the original paper. However, we have an important limitation. We do not know if they are together at the same time. If there are differences in phenology, because some quails are here and some of the ones are arriving. When the ones that are migrating arrive, are the other ones already breeding? So just looking at the frequency does not, of the different types does not mean that they are sexually active at the same time. So we had to narrow a little bit the window, and we thought it would be a little bit speculative to do it with the data that we already have. And also, the systems that we are using to capture, they are very efficient with males, but it's extremely difficult to capture females and chicks. We can only capture the chicks when they start flying, and the number of juveniles that we manage to capture is very small, and this estimate would have a big error. So yes, you're right, that is our intention, do something like a Hardy-Weinberg equilibrium, looking at with inversion, without the inversion. But we think that we need to be a little bit more precise to cut different time periods to see who is there at different times.
Yeah, I guess, well, in order to figure out what the reason of the disequilibrium, I mean, it's a good idea to somehow do a deeper study, but within the data you already have, you know, having A, B allele and in a single population and having typed homozygotes, I mean, you could check if there is random mating or not as a hypothesis, even if there are many causes for that.
Yes, you're right. We could make a guess, and I think that that's a line of work that we need to keep exploring. So that is something that we are discussing within the group, but it still needs to be further developed. I've been putting this figure before. In the Canary Islands, we have the 3 types, okay? We have lots of blue heterozygotes and few of these homozygotes. However, there are 2 islands that are very close to the mainland compared to the other ones. All the migratory quails appear in those islands and not any of the other ones. So it seems that from here, if we exclude those 2 islands, is Lanzarote and Fuerteventura, am I saying that right? The 2 closer to the mainland. In those islands, it seems that basically those are migratory quails that occasionally go there and they may disappear. But if we take those 2 islands away, that is what we would have. We would not have any black. We would have lots of blue and red. It seems that despite very few immigrants arriving, the chromosome without the inversion progresses within the population. We think that this could be suggesting that the red ones, the homozygotes, are not fully fertile, and maybe there is a reduced fitness, and that's why there is so many blue despite the few individuals that arrive there. So this is one of the areas that we are discussing, we're speculating, and it's going to be for future projects. So, but yeah, you're right, and that was our first intention, to look at the if there was an equilibrium between the frequencies of the different types. You're right.
Would you say that the inverted region of the genome is increasing the mutational robustness of the quails, in the sense that the inverted region can accumulate, or is accumulating, a lot of mutations? Most of them are synonymous substitutions, but at some point they might be preparing the ground for future adaptive mutations.
I mean, I like to speculate with that idea, but in fact, this is what we have seen with dogs and wolves. We did a very similar analysis with the collaborators in Sweden, looking at the genome of dogs and wolves. And basically we saw that in dogs that have a smaller effective population size, they are accumulating much more functional diversity, many more changes that affect the phenotype. And we all know the huge phenotypic diversity we see in dogs that could have been promoted by the relaxation of selective forces and the way we handle dogs. So at a different scale, we think that something like that could be happening here. We have lower restrictions on the quails, so lower, they do not have to migrate, they do not have to prepare the body for migration, and at the same time, the effective population size is smaller, that makes selection less efficient. So there are many of these slightly deleterious mutations that may remain in the population, could become fixed. So I think that yes, being summary, that in the islands, this could be accelerating the morphological diversity. I don't know, that could end up also increasing the genetic load, and problems of inbreeding could lead to extinction of some island lineages, like could have happened in the other Macaronesian islands. But we don't know, but I think that yes, this is increasing the diversity, the phenotypic diversity, for good and for bad.
So, thank you. We have several questions on YouTube, so in the interest of time, I'll try to pull several into one. So I'll group the questions of Joaquin and Caliwood. They both thank you for your fantastic talk. So then it's going to be a question about the, say, the implication of the inversions. So Joaquin was asking, for instance, if the reduced dispersal might be more beneficial in island population, and Caliwood was asking something more related to the pigmentation and the difference in coloration, if this having a lighter or darker plumage would be more advantageous, for instance, for the resident or migratory ecotypes.
Yeah, we don't have anything to support the the role of the pigmentation. It's true that the pigmentation has been related to immune system, but we don't have any evidence in that line. But what Joaquin says, it fits very well with the island syndrome, that lineages that colonize islands in birds is very, very common to lose migratory capacity as an adaptation for island lineage. It's not necessary to migrate because there is nowhere to go, kind of, no? So the investment on migratory potential can be reduced because it's not very relevant in that context. So I think that, in that sense, reducing the investment on resources for migration may be beneficial in island context, yes. I think that can be very appropriate in this context.
Hi, Carlos. It looked like you had a few other FST, like high FST genes popping up in some of the other chromosomes. Are those significant, or do you have any idea what those are? I mean, or in another
For
way,
example, this one here.
Sure.
There's a big peak.
Chromosomes as well.
Yeah. Some of them are very relevant. This thing here, these peaks here, is what led us to do this other analysis search, and we found this other inversion in chromosome 2. Some of these peaks, yes. However, there is one problem with this data. This has been mapped against the Japanese quail genome. So the Japanese quail and the common quail are expected to be very similar, but maybe the chromosomes and the genome sequence is not exactly the same. And it seems that if I'm, maybe Sarah could correct me if I'm, she's following from YouTube, but I think that this is just an artifact of the deficient mapping to this chromosome. And in fact, if you think for a moment on the second inversion that I was mentioning one inversion, but there are in fact two peaks, one here and one there. We know that it's in chromosome 2, but we are, at this point, we are not considering as two different inversions, but problems with the mapping because the reference is not quite perfect. So we have now a common quail genome that is not annotated, so we are discussing what would be the best strategy to do this analysis. We are not sure that this represents a different inversion. And yes, there are a few of these peaks that we have not really looked at carefully, and probably there are a few more of these inversions or big peaks that need to be carefully addressed. We have not been doing that yet.
I'm not sure if I have understood very well, but it seems that the absence of the inversion makes the birds prone or towards the migration, and it's also involved with sexual behavior like Don Juan de Marco, is it right?
Well, the absence of the inversion, let's see, that is the ancestral state, and yeah, quails migrate and have this extreme activity. It seems that the presence of the inversion is what reduces the flight capacity and makes the individual less fit for sustained flight. It seems that the inversion is associated with this reduced
the
capacity.
inversion is associated with the
Reduced capacity to disperse.
Okay.
Yeah?
I had almost a similar question that you have said that in Iceland, birds tend to lose the capacity of flight, or migration. And I was thinking about this not being associating with the inversion. So you have seen that the inversion is most common in the island, and you have seen a loss of capacity but are you associating the genes or something in the inversion with
it's just a correlation. It's just a correlation. You're right. We cannot prove that the inversion is responsible for that. You're right. Okay. Could be a coincidence, but the main point maybe would be the ones that are not in the islands but on the mainland. Yeah. That also around here in Seville or in southern Portugal, we have found also these dark quails with the inversion that show little differences, that seem to have very little dispersal. Yeah, no migration, and those are in the mainland. It's not the island syndrome.
Okay.
So it's a correlational study. We cannot prove that the lack of migratory behavior is within the inversion. It just seems to be, but we don't have a strong
Thanks.
the final conclusion, the final resolution, yeah.
We have time for a last one, if anyone. No? Okay. Carles, thank you very much, and thank you all for coming.
Thank you.